Hi everybody, welcome to our Vodcast on proteins and enzymes. In this one we're going to explore the organic molecule class known as proteins and get into a specific group of proteins known as enzymes. So without further ado. So we can organize all groups of molecules in life into four different classes, or groups rather. So we call these the main groups of organic molecules, and all organic molecules means is that these are molecules that are carbon-based, okay, just like we are. So we are primarily made of molecules that have a huge amount of carbon as an element in terms of backbone. And for all living things, these main molecules make up, or I should say, are made up of six main elements. We have carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. These six main elements make up all of these four groups. So let's take a quick look at the four groups. But like I said, we are going to really be focusing on proteins. Carbohydrates, that is one major group. Okay, these are things like sugars and starches. Lipids, those are our fats. Okay, there's that recognizable phospholipid molecule. Obviously, like I said, we have proteins. This is a big deal, okay? So proteins are very, very important, and we're going to learn that their structure is extraordinarily important to the function that they have. And the fourth class are nucleic acids. This is our DNA, and we are going to spend a lot of time with this group when we get into molecular genetics down the road. All right, so proteins. So remember how I said that there are six main elements that make up these molecules? Well, proteins contain five of those six. They contain carbon, hydrogen, oxygen, nitrogen, and sulfur. The only one they're missing is phosphorus. So you won't find any phosphorus present in our proteins. Okay, so there you have it. Proteins are made up of a subunit known as amino acids. There are 20 different types of amino acids that are present, and the way that we link them together, think of it like our alphabet, right? We have 26 letters. I can take those letters, arrange them in any order, in any length, and I create my words from that. So just like the way we do our words, if I take particular orders of amino acids, different amounts of amino acids, and link them together in a chain, I can create different proteins. So there are 20 distinctive different amino acids that build the proteins that we have. So just to show you a couple of quick examples, these are some of the amino acids. They have some very distinctive characteristics, but as far as we're concerned, we'll explore what these different ones are when we get into molecular genetics, but just to know, this is the big deal right here, is the fact that there are 20 different amino acids that exist that build our proteins. So these amino acids join together to form what's called a peptide bond. This is the special type of bond that holds each amino acid together to create that very unique order and length. And that chain, once I have that chain and it's twisted and it's roped into that very specific shape, now I have a protein. And I'm going to say this probably till you can't stand it anymore, but the shape of a protein really dictates the function. Kind of go back to when we watched Mad Cow, right? So Mad Cow, that prion, okay, was different from that normal protein because of its shape. And when you alter the shape, you alter its function. So what are some functions of these proteins? Proteins have a lot of different uses in us. We have about 100,000 different types of proteins. That's a whole lot. And we can kind of break all our proteins down into categories. So we have structural proteins. Big deal. Okay, they form our structure and make us what we are. They're embedded in the membrane, okay? Going back to what we just talked about with active transport proteins, right? Proteins, they're stuck in there so they can be channels and be ways for substances to cross that membrane. We also have and use proteins as an energy source, okay? They're not our primary energy source, but we do use them down the road to help us gain the energy we need. Hormones, okay? Hormones are our chemical messengers. They send signals from one part of our body to another to get something to happen, and many of our proteins are, in fact, hormones. Insulin is one. You might have heard of insulin because that is the hormone that regulates the blood sugar that we have, and so somebody who might be diabetic is either low on insulin or can't even create it. So that just might be one you've heard of, but we have a lot of different protein hormones. And lastly, and what we're going to focus on are enzymes. Enzymes are our biological catalysts. Okay, so this is a pretty key piece here. These guys are really, really important, and I can't stress this enough as to how big of a deal enzymes are. Our reactions don't happen without them. Okay, so let's take a little look at what enzymes are. So most of our enzymes are proteins. For our work, we're going to do that. They catalyze all of our cell reactions. So to catalyze something means that it's going to speed it up. So it allows for these reactions to occur at the speed at which we need them to. So they happen really, really fast. Now here's the thing. They promote the reactions. They speed them up. But they're there to kind of make sure it happens. They're not part of the reaction. They're just there to ensure that it happens. So for example, if I want to take something like, if I have substance A, chemical A, that I'm combining with chemical B, and as a result of their combination I get C, the enzyme will go here. It's going to speed it up and make sure that A combines with B to get C, but it's not part of the reaction. It's not used up in the reaction. It is there, and we can use it and reuse it, and it helps it happen really fast. So these chemical reactions that normally need high temperature, think back to diffusion box, right? When I raise temperature, molecules move a lot faster. And when they're moving a lot faster, they're more likely to bump into each other. So when they're moving a lot faster, the reactions are going to happen quicker. Well, in the grand scheme of things, we are really, really cold. And our molecules are not moving nearly fast enough for the reactions to happen as quickly as they need to. So what enzymes do is they make sure reactions are happening as quickly as they need to at temperatures that are what we consider to be a normal temperature, like our body temperature. So here's kind of the way that they work. Now you're going to want to get this diagram down. Okay, so you might want to pause, jot this down, and before we continue. This is known as the lock and key model. Okay, so the way an enzyme works is, and you don't have to worry too much about the coenzyme piece. All coenzymes do is they let a reaction happen a little bit better by making the enzyme fit what it's doing. So here's one where I have that idea of that A plus B gives me C model. Okay, so here's my enzyme, that very, very structured piece. When a coenzyme fits, notice my shape, okay? So the key is to notice the shape. Here's my substrates. Here's my substance A, here's substance B. So substrate is also known as the reactant of a reaction. Okay, it's what goes in. Now, what the enzyme does is it locks, okay? And the substrates lock at the enzyme substrate, create, I should say, the enzyme substrate complex. So the substrates, they go into what's known as, this area here is called, I'm going to bring that up, is called the active site. Very important piece, the active site. And the substrates fit because it's literally like a puzzle, okay? And if the piece or the substrate doesn't fit the active site, they're not going to combine. So it's a very distinctive, very specific fit that happens. Now when they're there, that lock and key, okay, now, hey, my substrates are really close together. That enzyme is going to, boom, link them together, and off comes C, my product. It's released. The enzyme can now be used again. We are going to do a worksheet class that showcases this as well. So if you're feeling a little funky on it, one, you can go back and look at it again and also see if what we do in class actually helps. Enzymes are very specific. Okay, we say we've got one enzyme, one substrate. Now, enzymes are involved, just like in that last slide, of building up molecules. Enzymes are also necessary to break them down. So not only do I use enzymes to do my A plus B gives me C, I also need an enzyme to, let's say, break C down into A plus B. But here's the kicker. It's not going to be the same enzyme. It's going to have to be a different enzyme that does that. So enzymes are extraordinarily specific to what they catalyze. And because they are proteins, and this is something that is a property of proteins, they are very temperature and very pH sensitive. As a protein, if alterations happen to these outside the normal, the temperature goes really, really high, the protein shape falls apart. It loses that really, really important shape that it has. And if it loses its shape, it can't combine with the substrate. So our active site, as I mentioned earlier, is part of the structure, and that can fall apart. It can be ruined by extreme alterations in temperature and by severe changes to pH, which we're going to spend some time talking about what pH is. And when a protein falls apart, okay, we consider it to be denatured. So this is a new term. So this is one that you're going to be needing to really understand. Denatured, when protein structure has altered so much that it renders the protein non-functional. Again, the structure of the protein dictates the function of the protein, and it is so, so important. Now, as I mentioned, we need enzymes because we're way too chilly. If our reactions, at the speed that reactions need to happen, okay, we would have to be somewhere over 200 degrees Celsius just for those reactions to happen if there were no enzymes. Obviously, that is far too hot for any of our proteins to stay together. They literally fall apart. So life can't exist at those kinds of crazy temperatures. And just as an aside, in case you're wondering what 200 degrees Celsius is, water boils at 100 degrees Celsius. So I'm talking hot. So with that being said, we are going to spend a lot of time going over what these are, do a little work with enzymes. We have a great lab called Pineapples and Enzymes where we're going to look at factors that can influence how an enzyme works, and take a look at some actual enzymes working in the lab. So have a great night. We'll see you in class. Take it easy.